Welcome students to this today's lesson. We are talking about biology paper 3. We are dealing with the food tests. For the purposes of this lesson, we shall be looking at three sets of foods that we'll be testing for their presence. So we'll be testing for starch. Other than starch, we'll also be considering proteins. We'll also be considering the test for reducing sugars. We'll also be considering the test for reducing sugars. Now, to start us off, from our theoretical analysis, we realize that there are those components of the body that are made up of certain structural components, and those structural components were made up of the amino acids, and they were also made up of the fatty acids and glycerol. So in that aspect, that is why we need to now to do experimental analysis to confirm that a particular food substance contain any of the three major food substances that we have. So we are going to be having, we have each and every one of us as a set of reagent. So we have a solution. For the purposes of this class, we shall be calling it solution L. Solution L is a mixture of different food substances. So far so good, we don't know the food substance that we are dealing with. So it is our work now to analyze using the reagents that we've been given. So we are going to test for starch, test for proteins, and also test for reducing sugars. So first of all, we need to know the procedure for testing these food substances. So, for example, when you're dealing with test for starch, then we consider the procedure for testing starch. So, as a student, it is very important that you have a correct procedure, because a wrong procedure will give you a wrong result. So, when you are testing for starch, the procedure number one that you are supposed to be dealing with is first you put 2 cm cubed of the solution of the food substance or the substrate into the test tube that you've been provided with. Then after you've measured 2 cm cubed of the food substance of the solution L, then you need to add 2 drops. You need to add 2 drops of iodine solution. You need to add 2 drops of iodine solution. Then you need to shake. Then you observe color change. You observe color change. You observe color change. Observe color change. So we are going to go with this so that we can be able to observe whatever color change. Then we will be drawing our conclusion. So we'll be doing our observations. Then after the observations, we shall be able to draw conclusions from this. We shall be able to draw conclusions from the results that will be given. So students, I want us to take our solution L, and you have within the rack that you are having, we have 3 test tubes. So you pick 1 test tube. Then for the purposes of this lesson, the test tubes have already been rinsed and they are clean. So you put about 2 cm cubed of the food substance. Put about 2 cm cubed of the food substance. So just that, about that is just enough for the purposes of being able to understand what is happening. Then to this food substance, we need to add 2 to 3 drops of iodine solution. So the iodine solutions have been given from the ones that are in the common table. So you'll be able to come and identify the iodine solution. Then you'll be taking 2 to 3 drops. 2 to 3 drops. Then you shake. Then you observe the color that is going to change. Observe the color that is going to change. Observe the color that is going to change. So you ensure you observe the color. So you don't put in excess. You don't put excess because sometimes when you put excess, you'll just be able to observe the color of the iodine, but not of the food that has changed, because it is not by virtue that all the time we must have that kind of food that you are testing. The food may not be there, so that when you put the excess of iodine, you'll be looking at maybe a blue-black color, but that may not be the color of the starch that has changed the food. So in this case, we can see that our color has not changed so much, but we have almost a green color. So it means that we only had traces of starch in our food substance. So in our observation, we'll be saying now the observation that we've made is that we had observation is that there is a green color. Then in our conclusion, we shall say traces of starch. So starch was there, but it was not sufficient enough to be able to change the color. So in ideal, when you observe a blue-black color, when the color changes to blue-black, then we expect our conclusion will be starch present. Our blue-black color will be starch present. Of observation, if we observe blue-black, then the conclusion should be that starch is present in the food substance. Now, having done so, now we want to consider testing for reducing sugars. Testing for reducing sugars. So, students, I think from the procedure, you've seen what I've done. So, I think I'll give you some 2 minutes to confirm with the food substrate that you have to ensure that you get at least closer to the result that I've gotten. Because we may not be very sure if the food substance that you have is the same that I have. So, kindly follow the procedure and perform the experiment. Follow the procedure and perform the experiment before we consider the test for reducing sugars. Experiments are done when you are standing. Normally, as a student in the lab, sitting is prohibited. What is the observation? Has the color changed? So the color has changed, but not so much. So traces of reducing of starch present. Traces of starch present. Now let's consider testing for reducing sugars. So we'll still use our solution L. We'll still use our solution L. So reducing sugars procedure. So said it is important that you have a correct procedure, because the correct procedure is what will give you the correct result. So procedure. Now, when you are testing for reducing sugars, you take 2 cm cubed. So you're going to add 2 cm cubed of the food solution, or of the substrate solution. For the purposes of the exam, you will not be permitted to say substrate solution, but you'll be naming the solution that you've been given. So you'll be saying solution L. So add 2 cm cubed of solution L to a test tube, to a clean test tube. Then you add 2 to 3 drops of Benedict solution. You add 2 to 3 drops of Benedict solution. Then you heat in you heat in a hot water bath, or you can as well heat it directly, but you take caution that it doesn't maybe splash onto you, So we are going to perform. We have the expected result that we need to consider. So we want to concentrate on the reducing sugars. Already we have the procedure. So what is remaining is for us to follow the procedure so that we can be able to see the kind of result and conclude whether or not we have reducing sugars present in the food substance. So we are going to put 2 cm cubed on a dry, clean test tube. We are going to add 2 to 3 drops of Benedict solution. So Benedict solution and copper 2 sulfate, sometimes they look more or less the same. So you have placed 2 cm cubed of the solution L, which is our substrate that we want to confirm if there is presence of reducing sugars. Now we are going to add 2 to 3 drops of Benedict solution. We are going to add 2 to 3 drops of Benedict solution. We're going to add 2 to 3 drops of Benedict solution. Then we are going to heat the mixture after you shake. After adding, then you shake. After adding, you shake. So you add 2 to 3 drops of Benedict solution to the mixture and shake. So you need to shake. Then after you shake, then you heat on hot water bath. So ordinarily, you are supposed to have a test tube holder. So you heat as you continue to do what? To shake. You heat as you continue to shake. Then you need to observe the order or the sequence of color changes. You need to observe the sequence of color changes. Even as you continue to heat. So as you continue to heat, you shake and you observe the color that has changed. Has any color changed? Has the color changed? It has changed to? Which color is that? Green. This is green, eh? So you continue, so you don't stop just by the first attempt. So you continue. Is there any color change? So the color is coming to something near brown, but it's not so brown as you expect. So it is persisting. Is it changing further? So it is persisting. It's persisting. So therefore, you need to make a conclusion. So if you make a conclusion, then you talk about the different color that has changed. You need to talk about the different color sequences that have changed. So you need to talk about the different color sequences that have changed. So you don't only talk about the last color that you've seen, but you talk about the first sequence of colors. So our final color is brown, but you need to start from the first color that you observed when you are doing the heating. When you are doing the heating, So when we see a blue color, so we started with a blue color, that is the color of Benedict solution. So if the blue color had persisted, then we would be saying that reducing sugars are absent. We'll be saying reducing sugars absent if the blue color had persisted. Then it changed to green. It changed to green, then finally to brown. So because the brown color has persisted, then we'll be saying that we'll be saying when we have brown, so it's either we see a brown color or an orange color. So when we talk about brown color observing, then we say that reducing sugars. So we talk about high amount of reducing sugars. High amount of reducing sugars. If we see a yellow color, if we had observed a yellow color, then we'll be talking about little amounts of reducing sugars. We'll be talking about little amounts of reducing sugars. But when the blue color persists, then we say reducing sugars are absent. When the blue color persists, then we say that reducing sugars are absent. Reducing sugars are absent. So it is important when you are giving your answers, you give your answer in terms of the color sequences. So you talk about that the color change from blue to yellow to brown. From blue to yellow to green to brown. So you talk about the sequence. So you don't only look or give conclusion based on the first or the last color that you have observed. So it is important that you take note of that. So it is your turn now to confirm with your with the food substance that you have if you are going to get the same as you heat, you don't have the color changing. As you observe the color changing. As you observe the color changing. So observe the color changing. So, full session of looking at the reducing sugars. Now we want to consider the testing for presence of proteins in any food substance. And first we realize that proteins are the building blocks for proteins are known as amino acids. They are known as the amino acids. Now, proteins are generally used for body building, that is increasing in mass. And most of the cells and even the entire bodies, we have the components of what? Of proteins, be it for plants or even for the animal body. Now, it is very essential, but it should be noted that sometimes proteins are also used as a source of energy, especially during starvation. So we want to test if the proteins are there. So the reagents that we'll require in testing for proteins, the reagents that you'll require will require sodium hydroxide. Then we'll also be requiring copper 2 sulfate. We'll also be requiring copper 2 sulfate. You're also requiring copper 2 sulfate. So we realize when you are testing for starch, our requirement is iodine solution. When you are testing for reducing sugars, our reagents are Benedict solution. So sometimes the exam will come, but you'll not be told to test for proteins or test for starch. You'll just be given reagents and told to test for the present or the food present. So here we'll be needing sodium hydroxide as well as copper 2 sulfate. So the procedure you put 2 cm cubed of solution L in a dry test tube. Then you add equal amounts of sodium hydroxide solution and shake well. Then add equal amounts of copper 2 sulfate. Add equal amounts of copper 2 sulfate. So this one may not be equal, but just few drops. Few drops is just enough for you to get the required result. So after adding, you also continue to shake well. So you should be, the main thing here is first, you put the food substance, then add sodium hydroxide, then after that you shake so that the mixture mixes well. Then it is when now you add your copper 2 sulfate. So to the remaining test tube, you put 2 cm cubed. The remaining test tube, you put 2 cm cubed over put 2 cm cubed over the food or solution L. Then we are going to add 2 to 3 drops, equal amounts, not 2 to 3, but equal amounts of sodium hydroxide solution. We're going to add equal amounts of sodium hydroxide solution. Then we are shaking. Then we shake until the mixture mixes well. So when you are shaking any reagents in the laboratory, ensure you don't shake longitudinally. You shake in the transverse section. So if you shake longitudinally, if it is a chemical substance, then it will may interfere with your fingers. So you ensure you shake it in such a manner that it does not affect your body. So the substances are not going to be in contact with your body. So after shaking well, then you are going to add a few drops of copper 2 sulfate. So few drops, then you shake. Few drops, then you shake. So we can see that the color has changed to purple. The color has changed to purple. So observation, color changes from blue to purple. So observation would be color change from blue to purple. So blue is the color of copper 2 sulfate. Then the purple. So the presence of the purple color, we say now proteins present. Proteins present. When the blue color persists, if the blue color persists, then we say proteins absent. When the blue color persists, we say proteins absent. So if the blue color persists, then we say that the proteins are absent. Proteins are absent. So kindly confirm with the liquid that you have if the colors have changed or the colors have persisted. Kindly confirm if the colors have changed or the colors have persisted. So kindly ensure that you use the reagents that you've been given well. Ensure you understand or you know the reagents that you're using before you apply them. Ensure you know the reagents that you've been given before you apply them. So after you added the sodium hydroxide, you shake until the mixture mixes well. That is when you add few drops of copper 2 sulfate. Sorry, that's when you add few drops of copper 2 sulfate. So you shake well until the mixture mixes. Then you observe the color change. Then you record the color that has changed. So just to sum up or to summarize whatever we've learned today, we were talking about food test. That is more or less a compulsory question in paper 3. Paper 3 consists of 3 questions. It consists of 3 questions. So one of the questions that may be there in question 1 will be part of food test. Will be part of food test. So in terms of food test, there are several foods that we can test for. So we can test for starch, we can test for proteins, we can test for reducing sugars, vitamin C. That you are given, try to know